it's my turn, and I would love to discuss with you more about shotgun lipidomics. So I guess you can see my presentation, and yeah, so I had great introduction here from the previous speakers, so I can speed up in some parts of my presentation. Let me give you a brief overview. So I would love to discuss here sample preparation first with you, and then identification of lipid molecules. You heard already several things about that. So there's difference maybe between low and high mass resolution analyzers. And very important point to get to accurate and really correct annotations. You have to consider here the type 2 overlap. I will show you that in a minute. Shortly, data processing and some resources for general lipidomics. So as you heard before from Maria, and then also from others, usually a lipidomics workflow has 3 steps. First, extraction. Then we have here either an LC or a direct approach coupled to a mass spec. Yeah, usually now it's not working. And there it's key to come to correct identification of lipid molecules at different levels. And finally, quantification. Yeah, that is not topic of this talk now. This will be tomorrow's talk. I would love to discuss more of quantitation by MS methods. Here I would love to discuss first here sample preparation. And yeah, what do we need? We usually have aqueous and materials, and if we have solid materials, we have to homogenize these materials. And typical requirements are here a cell protein from 100 to 200 microgram cell protein. This could be also lower depending on the instruments you use or dilution factors. Typically for plasma, we use, for instance, 10 microliter, or tissue 1 to 2 milligram wet weight. These are typical numbers applied to lipid extraction, and methods used here are, for instance, the Bly-on-Dia method. We prefer that in our laboratory. You know that you have aqueous volume. Yeah, this could be a diluted plasma or something like that. You add here a chloroform methanol mixture. In the first step, you have no phase separation. So you have time to incubate or to extract the lipids from binding proteins and from the matrix or biological matrix. And then maybe after an hour you add here part of chloroform, part of water to induce phase separation, and you recover in this case the lower chloroform phase. Yeah, and this type of extraction methods, same here with the MTBE method. You know probably they recover a broad lipid spectrum. Yeah, and here for the MTBE, it's principle the same. Yeah, you have first no phase separation in the first incubation, and then you induce a phase separation. But in that case, you recover here in the MTBE, the organic phase is the upper layer. Yeah, in the Bly-on-Dia or Feucht methods, chloroform, you have to recover the bottom phase, which is clearly maybe some disadvantage. Yeah, because you have to cross here usually you have here a protein layer or precipitation layer. You have to cross that and do that carefully to not to recover here a pure organic phase. Same here with the MTBE, or not. You have not to cross that barrier, so it's much easier. And to recover these organic phases, you may use here a pipetting robot. Yeah, and that is quite nice to do that in a high throughput. And Christian mentioned that also in the other setup. So how do we identify lipid molecules by mass spec? Maria told you already something about that. And let me start a bit historically. And that was actually the study which I, at least for me, was some kind of birth of lipidomics. This was a publication by Britta Brügger and colleagues, where they did nano electrospray and really tried to get a lipidomic or lipid profiles from tiny amounts, of material. And here you see a list of lipid class specific fragments. So they used at that time a triple quadrupole instrument. So these were the first instruments used for lipidomics. And yeah, you find here negative ion mode fragments and positive ion mode fragmentation. I will show you some examples in a minute. So these are, as you heard from Maria, low mass resolution instruments. So usually you have mass windows of mass resolution, unit resolution. So it's 1 Dalton usually. And Maria explained already the setup here of a tandem mass spectrometry. In this case, we have 3 quadrupoles aligned, and we ionize usually with the electrospray ionization, and we can run different modes. Yeah. So, for instance, we can analyze product ion spectra. That is the first quadrupole is used as a mass filter, selecting one particular mass unit. So, and these ions are entering the collision cell. They fragment, and you can scan in the third quadrupole, so you will get all the fragment ions. You will see them. The other way around, for instance, which fragment ion you expect, then you can yes, the third quadrupole is set on a specific M/Z, and you scan in the first quadrupole. Now this is called a precursor ion scan. And if you know that your particular lipid of interest loses a specific fragment, you can do type of neutral loss scans, which means both quadrupoles scan with a certain mass offset. Yeah, this is called a neutral loss scan. And Maria explained you already here selected reaction monitoring. You just select in the first and in the third quadrupole specific fragments or precursors. So this is a PE molecule in positive ion mode, and if we subject this PE molecule with their M/Z value of 718 to fragmentation, we will generate in positive ion mode here a neutral loss. And this is 141. And if we see the fitting fragment, the matching fragment, we can of course not annotate the acyl chains. Yeah, we only can annotate here the sum of these acyl chains, because we cannot differentiate, of course, these chains, and we call that here lipid species level. And here comes some kind of assumption into the game, and this is in that case we assume that only even numbered carbon chains are present. I will show you in a minute why we need that kind of assumption, because there are isobars present, which will I explain also on the next slides. So this is lipid species level. If we do, for instance, negative ion mode with the same lipid, we see another kind of lipid class specific fragment. In this case is 196, and that is specific for PE. But in negative ion mode, we typically see also fragments from the acyl chains. Yeah? And here we have a 16,0, it's 255, and then 18,1, it's 281. So having all these three informations, we can annotate at molecular lipid species level. So we know this PE species has an palmitic acid and the oleic acid. So but it's difficult to say where at which position this acyl changes are linked. So we use typically here the underscore. Yeah. If we know the SN position of the acyl chains, we use this slash, but most methods are not able to figure that out. That is really sophisticated analysis. You heard already, you have to calibrate your instrument or use other methods to really come to that level of information that you can can prove the SN positions of your acyl chains. So that is another example. It's a PC molecule, also in positive ion mode. Here it's a very specific fragment. It's a 184 fragment. It's a head group of a PC molecule, and in that case also it's here lipid species level. You need also this assumption. And if you use that fragment in a precursor ion scan, you have also a second type of lipid class you will get from a crude lipid extract, and it's not here only PC generating that fragment, it's also sphingomyelin generating the same fragment because both contain this phosphocholine head group. So you will see here from this precursor ion spectrum that PCs have even M over Z values, and here sphingomyelins, they come are displayed with or they have odd M over Z values. So you can easily imagine that you have some kind of isotopic overlap, which you have to deconvolute, and I will show you that also on the next slides. So it's not so straightforward just to pick here the intensities and have with that here accurate values. Yeah, you have to really do some data deconvolution to have really exact concentrations. So if you use here negative ion mode, yeah, same as with the PE, you have here lipid class fragment. It's here a neutral loss. If you have here a formic acid adduct for the PC, for instance, and also acyl chain fragments. So you can here again annotate at the molecular lipid species level. This is a spectra of a PS. Yeah, from Steve Blanksby's group, and here you see you have not only these acyl chain fragments in negative ion mode, but you also have lyso type of fragments, which you can use for to get more evidence that your annotation is correct. So you see here rich fragment rich spectra in negative ion mode, and you may combine that of course with positive ion mode data, where you have different fragments, and also here of course also minor fragments also for the lyso type of fragments, but here it's a neutral loss, which is a lipid class specific for the PS. So at the end you could use both positive negative ion mode and really come to a justified to a proven annotation of your lipid, of your MZ feature you observe maybe in an MS mode, but frequently it's also not so easy, because if you use direct infusion, you have quite crowded. We heard it also in the previous talks. There are a lot of different lipids present at the same mass to charge ratio. And Xianlin Han is pioneering that field, and he developed here use of various different scan modes to figure out the composition of the whole lipidome. And you may use here for instance for PI 241 scan to see all the PIs. Yeah, this is a minor contribution here. You see in that mouse liver extract, you have here for the serine or for the PCs, you can apply different scan modes, and you combine these information with the fatty acid scanning. Yeah, so for the head groups present, so the lipid class present at a certain mass, and you can combine it here with the acid data, and in that way you may really come to molecular lipid species data for your sample. So let me summarize the principles of low mass resolution tandem mass spectrometry. So I have to say, with the single mass spectrometry, you are not able to do that type, or to identify actually lipid species by flow injection or direct methods. So you have to use here really tandem mass spectrometry, and you have to evaluate the different lipid class fragments, which may be either direct fragments from the head group or neutral losses. And if you want to reach that molecular lipid species level, you have to see the fragment from the acyl chains. So that is really necessary to get that type of information. So now I would here switch to high mass resolution, and first discuss these terms with you. What is an isomer actually? So here are different isomeric species for PE and PC. And you see we have variations here in double bond positions or chain length. And for instance, we have also isomeric PC and PE species. So it means they have really exactly the same molecular formula. Yeah. So you cannot separate these molecules just by MS. Yeah. For so PC and PE are isomeric, so you have just 3 additional carbons here in the acyl chain, and they are missing here at the amino group. But you already learned that we can use here tandem mass spectrometry, yeah, because we get different fragmentation. Yeah, so we get here a neutral loss of 141, and here we get a fragment of 184 for the PC in positive ion mode. So it's easy to differentiate actually these 2 isomeric species by tandem mass spectrometry. Or you have also seen separation is possible. So isomeric species, not simply the mass can differentiate, but we can use, for instance, fragmentation to differentiate these molecules, or also here we can differentiate the acyl chains in negative ion mode. It's harder to differentiate here double bond positions, but you will hear more advanced techniques in the coming days. So what is in contrast to isomers? It's exactly the same molecular formula, an isobar. An isobar has not the same molecular formula. And here you have, for instance, a PC D-acyl species and a PC which has an ether bonding here in the one position. And you see this is 33 carbons, and here we have 34 carbons. And if you look at here the mass in the positive ion mode, we have here same nominal mass, but we have different decimals. So these have not exactly the same mass, but the same nominal mass, and of course here mass spectrometry is capable to differentiate those, but you need a high mass resolution. Yeah, sufficient mass resolution is necessary to differentiate this small mass difference, and you see here we need a mass resolution, yeah, let's say about 40,000 to separate these 2 isobaric peaks. And therefore we need that type of assumption if we annotate here our low resolution mass spectra, because you see we can not with the triple quad, we can which says unit mass resolution, which means it could differentiate 746 from 747 easily, but not that type of small mass difference. Yeah. So for triple quad analysis, we typically use that type of assumptions that we say we have only even carbon number. So we would annotate that based on that assumption, that peak as a ether species. But honestly, that is just an assumption. It really depends on your biological material, whether that is the majority of that peak. We know there are a lot of odd chain numbered carbons, for instance, in cell culture samples. So that is the beauty of high resolution. You can really differentiate these bond types. And yeah, as already Christian mentioned, high resolution mass spectrometry is frequently coupled here with the device here from Advion. It's this nano electrospray chip based device, and it's introduced by Andrei Shevchenko's group. Yeah, several years ago. And initially they used a QTOF instrument and performed similar experiments as Xianlin Han. But the beauty of a TOF instrument, of course, is you get the whole fragment spectra at once, and it's not so tedious with as with the triple quadrupole instrument. And so you, but in principle, you can here combine data from a head group scan. So you define which lipid class is present, and you combine that with the fatty acyl scans, so you know which acyl combination is present, and this allows you to really annotate on a molecular species level. So nowadays, more and more here, higher mass resolution instruments are used for lipidomics. Christian mentioned already here Orbitrap instruments. It's a Q-executive, so you have a quadrupole and an Orbitrap detector here in this hybrid instrument. And these instruments allow really nice mass resolution. And for instance, you can use that here for triglyceride analysis. And this is here a human adipose extract. It's Dominik Schwutke's paper here. And you see you can nicely annotate at the lipid species level, but you can also use tandem mass spectrometry to figure out the acyl combinations, because these triglycerides, they lose acyl change as a neutral loss, and you can combine this information to have this molecular species level. So it is also nice to have here high resolution for your tandem mass spectra. For instance, if you want to differentiate this kind of fragments, so for this PS species, you have, of course, an acyl fragment for the 18-0, but also the docosahexanoic acid. And for these highly polyunsaturated fatty acids, it's known that they lose CO2 during fragmentation. And you see you get here some kind of isobaric fragment. Yeah. For the triple quad, you will never be able to differentiate that, but with a high resolution, you can differentiate this fragment from the stearic acid fragment. So let me summarize high resolution mass spectrometry. And here you use more the accurate mass, yeah, to identify your lipid species. But we have to say that is not usually not enough. Yeah, you should really use maybe both polarities or tandem mass spectrometry to be sure for of your annotation. And Maria pointed that already out. It's nice to have this high mass resolution to resolve isobaric interferences, for instance, ether and diacyl species. That is straightforward with a high mass resolution. So what is really not an easy thing in lipidomics, and this is the so-called type 2 overlap. And this is resulting here from double bond series. And what do I mean with that? Let's here have a look at the PC36-1. And this is an isotopic pattern here of it's just a simulation of a PC36-2. And you see that you have the +2 isotope. It's present in about here, yeah, around 15% related to the monoisotopic peak. And this comes mainly from the C13 content. You all know that carbon 13 is present about 1% in nature. So with the higher mass, we have a higher contribution of this M+1 and M+2 isotopic peaks. And if we have a look here at PC36-1 and PC36-0, the +2 isotope has quite a similar mass than the monoisotopic peak of the saturated PC species. You see, it's only a mass difference here for about 9 millidalton. And this is a difference, of course. Again, the low mass resolution cannot discriminate that type of mass difference. And also TOF analyzes with a, yeah, it's hard. Yeah, they usually can also not discriminate that. You need orbit traps or higher mass resolution to really solve that type of overlap. And clearly, yeah, it's necessary to get some correction or differentiation of that type of overlap. Otherwise, you have huge false misidentification and also quantification of some peaks. So here, this is from ancient times. Yeah, it's a triple quadrupole type of study, and there we used the precursor ion scan of 184, which means we get here sphingomyelins and PCs. And in this experiment, we just spiked here, for instance, this sphingomyelin M/Z 731. And you see if we spike it, add it to, I think it was a plasma lipid extract, we get an increase also of this PC species, despite this PC species was not added in the experiment. So we need some kind of correction for that. And we did correction just based on the relative isotopic abundance, which you can calculate with the isotope calculator. And if we do that in a stepwise manner, you see, despite we add here this sphingomyelin, we get more or less constant ratio to our internal standard for that PC species, just differing by 1 mass unit. And this is very important to avoid false identification and also quantification. For instance, this PC38-2, it's present only at a minor concentration. You see that here after correction. But if you omit correction, yeah, you would have here a substantial amount of that species calculated. So that is a really important step, and you have to keep that in mind for direct methods, and also for if you use HELIC, you have also co-elution of all these species, but also in your reverse phase methods, you could have some kind of overlap, because in tandem mass spectrometry, you always have an isolation window usually of 1 Dalton. So you co-isolate usually also some other species. So you may have also these type of isotopic overlaps. So with higher mass resolution, you may be able to resolve this type 2 interference. Here it's an orbit trap used here at 140 K mass resolution, and then this is at 200 m/z, and this is sufficient, for instance, to resolve here the LISO-PC 18.1 and 18.0. Yeah, this in this experiment, Marcus from our lab did a spike experiment. He added here the 18.1 in increasing amount, and you see the peak is rising, but we get some kind of separation of the saturated LPC species. With this orbit trap analysis, you get a decrease of the mass resolution for higher MOZ values. And so you see, if we go, for instance, here for the sphingomyelin species, yeah, we have not really good separation. Yeah, it's more or less one peak. And for the TG species, we at 900 MOZ, we get no separation. Yeah, so you have to consider that kind of overlap also with higher mass resolution, or unless you have really sufficient mass resolution, maybe the double, the high frequency orbit traps, they can resolve also here the TG species at that level. So you need no consideration of that type of interference in your peak identification. So let me summarize that part. You have seen it's really based our annotation and identification on the type of analysis we use. If we use here low mass resolution, we may need some assumptions in our annotation. If we have higher mass resolution, we can, for instance, resolve that type of isobaric species. And if we want to annotate at that kind of level, yeah, we need justification of these acyl fragments. And I have to say, you should see both of them, yeah, not just one. You should really have the evidence of both fragments. And positional analysis, it's quite complicated. I will discuss a bit during quantification tomorrow. And this type of really chemical resolved information usually it's not reachable with the mass spec approaches we use today typically. So you heard already, and I'm really delighted to see that Bruker is applying that type of nomenclature. So it's highly recommended that we have a common language which reflects here the level, the hierarchical level. So we can really recommend that to you to use this kind of nomenclature to annotate your data. And now I would love to discuss the remaining minutes a bit data processing. How do we get the data out of these direct methods? And let me first start with our quite old method. We used here a Neolinks tool from Waters. It was a triple quadrupole instrument. It's a Quattro Ultima, but it's still in operation almost 20 years now. We run that system, and we use here, we just combine spectra here from the TIC at half height of the injection. So it's flow injection. We inject here the sample. You see the total ion current is rising and declines, and we summarize these spectra in that window. Have combined spectra, they were smooth, were centroided, and that data were exported to Excel macros, and there we do the de-isotoping, as a really very important step, and this is based on isotopic pattern which calculated theoretically, and then but we do also quantification, and this is a topic of tomorrow. We use some calibration lines and to really calculate here mole of these species. So that is because at that time we started that no other programs were available. Nowadays, we heard already also from Christian that, for instance, Lipid Explorer, which is developed in the Shevchenko lab, this is a program very suitable for high resolution mass spectrometry spectra, and this is a script-based software, and you will hear also, I think, the coming days from that in more detail. That is applied by several labs here for flow injection or shotgun data. And we use here a software from Christer Ising's lab. This is Alex workflow. We use it for our orbit rep data, and there it's yeah, you can extract data from these experiments, and we programmed here some Excel macros also to deconvolute this data a bit more. So you see, basically there is no perfect software piece. Maybe the Lipid Explorer could be recommended here, but yeah, you have to try and really make your own picture from that. So finally, some resources here. I want to mention that here the Lipidomics Standard Initiative page has some collections of these lipid class-specific fragment ions. Yeah, we discussed already here for the PC, for instance, this 184, and also for other lipid class, positive ion mode, negative ion mode. We have here a collection. And we have also for the molecular lipid species, yeah, we mentioned that you here have two fragments yeah, for the individual acyl chains. So you should see both of them. Yeah, you see also in positive ion modes these lyso fragments, but they are frequently quite minor fragments. So in negative ion mode, it's much more easier, straightforward with these fatty acyl fragments. Yeah, you can have a look at that. We have also a collection of typical occurring lipid isomeric overlaps. Yeah, for I mentioned already PCPE. Yeah, this is just 3 additional methylene groups in the acyl chains for the PE. Or yeah, there are also some others present. Maybe not be a full collection. So if you have some suggestions, please send us an email. And we have also a list of typical isobaric overlaps. I mentioned already here the type 2 overlap. Yeah, there are also others, for instance, in positive ion mode. I will discuss tomorrow here. If you have sodiated and protonated adducts, you have a very small isobaric difference, which could interfere your spectra. And finally, there is also a potential source of misidentification. It's in source fragmentation. Maria mentioned that already. For instance, ceramides, they typically lose here water in the source. They can lose that. So, and that may lead to misidentification. But there are also several others. For instance, cholesterol ester, they lose here the acyl chain to generate the cholestadiene ion or the others, triglycerides. They lose fatty acid and generate the diglyceride. So you should check whether that, or to which amount you see this, which intensity these in-source fragmentations have in your instrument. You could check that with a standard. Otherwise, you will maybe fail in misinterpretation for especially if you use direct methods. Yeah, with the LC methods, you will have different retention time, of course. So at the end, just one word to potential applications, and because I like this type of applications very much, and you could use that shotgun methods easily to follow here the metabolism. And here it's one part of the major pathways of glycerophospholipid metabolism. Aoki told that already to you. And you can introduce here, for instance, a labeled ethanolamine to label your PE species. Yeah, so you will have not the neutral loss of 141, but 145. Or the choline, yeah, if you use a D9 labeled choline, you will have not a 184, but a 180, 193. And for the liver, you have this particular pathway, you have a methylation of your PE to the PC. So you are basically can follow that if you're here measure PC species with a 188. Yeah, this means that the ethanolamine newly synthesized is converted by methylation to the PC. And if you do that, for instance, in mice, yeah, they are labeled just by intraperitoneal injection of these two precursors. You see, this is a spectrum of the unlabeled PE species, and here you see the labeled PE species. Yeah, it's a Kennedy pathway. And if you have a look at the PC species, you see that they are quite different, whether the PC is derived here from direct choline incorporation, from that PE conversion. It's a methylation pathway. This is a D4 PCs. You see they are much more unsaturated compared to the PC species derived here from choline. So this is quite a nice application, and it's straightforward. So yeah, let me acknowledge here the people in the lab, especially Marcus. He did a great job, or does a great job still here. He's still here in the lab. Chris Rising, Chemacros, close collaboration partners. Thank you for your attention. These are our funders, and I would love to invite you here to Regensburg. We hope that we can have this meeting. It's a first ILS conference, and it's a joint with the Lipidomics Forum. We hope we can have that as face-to-face here in Regensburg in October, and it would be great to see you all there. So, and with this, I'm at the end, and I'm happy to take your questions.